Good evening. Today I will be discussing the effects of chlorinated disinfectants on the bacterial activity of Escherichia coli. To begin, I would like to share a little bit about myself. My name is Carys Johnston and I'm currently a senior at Chatham High School. Following graduation, I plan to attend Ferrum College where I will be majoring in pre-professional health as well as playing collegiate soccer. I began this research because of my personal struggles with skin sensitivity. As a person with eczema, I found that when I'm exposed to heavily chlorinated water, I experience painful symptoms such as rashes, itchiness, and the redness of skin. I also decided to study this topic because of my interest in microbiology and healthcare. It is interesting to learn about the properties of bacteria, specifically E. coli, because it can be easily spread and can cause severe harm to human health. That is why it's crucial to research and develop effective and safe disinfectants that will eliminate E. coli but will also be safe for public use. Before I performed my experiment, I read several studies to strengthen my understanding of the topic. A study explored that common environmental bacteria subject to stress conditions, such as starvation or temperature change, are highly likely to develop resistance properties. These properties protect the bacteria from being killed, whether that be with an antibiotic or a disinfectant. Staphylococcus aureus and E. coli are common examples of these bacteria. Researchers are trying to reduce their occurrence, specifically in healthcare and pool settings where more problems occur. Many studies have centered their research around how disinfectants will affect Staph aureus. This is because of the need to reduce the common infection that Staph aureus causes, known as the staph infection. Because of Staph aureus' popularity in disinfection studies, other bacteria such as E. coli are not widely tested. According to researchers, these two bacteria have similar growth phases and resistance rates, and therefore it is concluded that E. coli can be used in disinfection studies. I also reviewed studies concerning the disinfectants themselves. Chlorinated disinfectants are known for their innate ability to eliminate all of the bacteria on an applied surface faster than a non-chlorinated disinfectant. However, because of the recent overuse of chlorine solutions and the development of bacterial resistance, they have not been as effective. It was discovered that in order for the disinfectant to effectively eliminate the bacteria, they have to be used at much higher concentrations. This implies that chlorinated disinfectants are being overused because they fail to kill bacteria due to the development of resistance properties. While reviewing the literature associated with my topic, I discovered that there is a gap in this field of research. Many studies focus on the effectiveness of disinfectants on drug-resistant Staph aureus with minimal research on other types of bacteria. There is also limited research on the efficacy of chlorinated disinfectants on these bacteria. Therefore, researchers suggest that future studies focus on researching and developing an effective disinfectant that will eliminate both resistant and non-resistant bacteria. Due to the limited research on this topic, it has led me to the question, which chlorinated disinfectant among sodium chloride, sodium dichlor, and sodium hypochlorite will be the most effective at eliminating Escherichia coli? Before performing my experiment, I also developed my hypotheses based on previous literature I have explored. My null hypothesis states that if the chosen chlorinated disinfectants are tested on Escherichia coli, then there will be no difference in bacterial growth among the treatment groups. My alternative hypothesis states that if the chosen chlorinated disinfectants are tested on Escherichia coli, then there will be a significant difference in the bacterial growth of each group. I developed this hypothesis based on previous studies which noted that sodium hypochlorite is the most effective disinfectant when tested on Staph aureus. The independent variable was the type of chlorine-based solution. The dependent variable was the growth of E. coli, and to accurately compare how the disinfectants will affect the bacteria, a control solution of just distilled water was used. In order to ensure the validity of this experiment, the nutrient agar, temperature of incubation, and time in the incubator remain constant for all treatments. I began this experiment by obtaining my chlorinated disinfectants. 2 of the 3 solutions had to be diluted into distilled water, which is seen in the picture on the left. I then gathered the petri dishes, the bacteria, and the swabs and began plating my dishes. I had a randomized sample of 30 quadrant petri dishes totaling 120 individual samples. Each treatment group had 30 dishes, providing more accurate results. In the top right photo, I used a sample of E. coli to swab the petri dish side to side while rotating the dish in order to ensure a uniform layer of bacteria was created. While swabbing the bacteria, the zone discs were soaking in the disinfectants. In the bottom right photo, with sterilized forceps, I placed the chlorine-infused disc into its corresponding quadrant and then closed, sealed, and inverted the dish. After this process was repeated with each treatment, I placed the dishes into the incubator at 37 degrees Celsius for 48 hours. Once the incubation was complete, I recorded the zones of inhibition in millimeters using a ruler and then placed the data in a spreadsheet. I then conducted a statistical analysis using the data classroom software. To visually display my raw data, I used a scatter plot to represent the results of each treatment group. The graph shows the zones of inhibition in millimeters for the different treatments: sodium dichloride, sodium chloride, and sodium hypochlorite, as well as the control. It also identifies the average zone of inhibition for each treatment and the variance among the samples. As outlined in the red box, sodium dichloride produced the largest average zone of inhibition with a total measurement of 36.6 millimeters. The variance among this group was large, meaning that the zones of inhibition within the samples were not consistent. However, because of outliers, this variance has been skewed to represent a wider set of data. This was most likely due to human error while in the lab. Sodium hypochlorite was the next largest inhibitor with an average zone of 12.2 millimeters. Compared to the sodium dichloride solution, this treatment was not a large inhibitor. However, when compared to the remaining solutions, this treatment was significant at inhibiting the bacterial growth of E. coli. It is outlined on the graph that on average, sodium dichloride was not an inhibitor of the bacterial growth of E. coli. Regardless of two outliers, this treatment produced no zones of inhibition, making the average measurement less than 0.5 millimeters. This solution produced similar results to the control solution of just distilled water. Based on the raw data collected, a one-way ANOVA test is an appropriate hypothesis testing method for my experiment because I'm comparing the average zones of inhibition among 4 different treatment groups. After performing the ANOVA, it was revealed that at a significance level of 0.05, there is sufficient evidence to conclude that sodium dichloride is the largest inhibitor of the bacterial growth of E. coli with a p-value less than 0.01. This means that my data is statistically significant because sodium dichloride had a large effect on the growth of E. coli, and therefore I rejected my null hypothesis. The sodium hypochlorite solution held similar results to this solution because it also produced a zone of inhibition. It was just not as significant. Sodium dichloride's large performance may be due to its composition, specifically the amount of available chlorine in the solution. 58% of sodium dichloride solution is available chlorine, while sodium hypochlorite is only 48% available chlorine, and sodium hypochlorite is 36% available chlorine. The picture on the right shows the zones of inhibition from the 4 different treatment groups. The sodium dichloride solution is in the top right quadrant with the largest zone of inhibition. The sodium chloride solution is in the top left quadrant with no inhibition, and the sodium hypochlorite solution is in the bottom right quadrant with the second largest zone of inhibition. The bottom left quadrant was the distilled water treatment, which was labeled as the control and also produced no inhibition. The results of my study concluded that sodium dichloride and sodium hypochlorite may be used as inhibitors of the bacterial growth of E. coli. It is necessary to continue researching and developing additional disinfection methods as E. coli can develop resistance to our particular disinfectant. Ultimately, the development of effective disinfectants will be beneficial in fighting bacterial infections that are caused by this bacteria, leaving a safer environment for society. Current disinfectants are failing to eliminate E. coli and are also causing skin irritation because of the high concentrations being used. Therefore, sodium dichloride and sodium hypochlorite can be used to effectively eliminate E. coli and should be tested in epidemiology studies in the future in order to determine their effects on skin health. When reviewing literature and planning my methodology, I attempted to perform my experiment on Staph aureus, as many previous studies focused their disinfectant testing on this bacteria. However, because of restricted access to this pathogen, our program was not able to purchase it for the use of my experiment. While this did alter my original plans, it led me to discover a much bigger gap in the field, as there is minimal testing of disinfectants on E. coli, which holds similar properties to Staph aureus. There were also inconsistencies with how I obtained my chlorinated disinfectants. 2 of the 3 solutions came from the same company, while 1 was provided by Governor School, which was obtained from a different source. This could have altered my results, as the solutions were created and manufactured from different companies and may also have other additives in them. Future research should focus on expanding the types of disinfectants that can be used to eliminate E. coli while increasing trial number and sample size to provide accurate results. Throughout the process of my senior research project, I have learned the importance of time management skills. While it is not always easy to stay on task and keep a positive attitude, I have noticed that if you effectively manage your time, the research process will be much smoother. I have also taken into account the potential harm that these bacteria can cause, which encourages me to take more precautions in order to avoid harmful infections that can be caused by this bacteria. Finally, I would like to give a special thank you to Ms. Long for her assistance throughout the research process, Mrs. Dillon for her help in the lab, and Mrs. Bowman and Mr. Reese for allowing me to carry out my research. That concludes my presentation. Are there any questions? Yes, Mr. Taylor.
So on your zones of inhibition, where you placed your discs in your Petri dish, can you explain exactly how you measured your zone with relation to that disc?
Mr. Taylor is asking how I measured my zones of inhibition with relation to my discs. So because my Petri dish was separated into quadrants, there was a smaller circumference around the dish. So I measured the diameter across the dish as it was not completely symmetrical, so I used the widest area that was in. Yes, Mr. Taylor.
So what was your criteria for saying that the zone did not actually have bacterial growth? I mean, was it just a certain density of bacteria, or did you look for no growth at all?
Mr. Tellerico is asking how I identified if the zone of inhibition actually did have growth or not. I identified if it had growth based on if I could visibly see a difference between the area that was supposed to hold an inhibition. So if I did not see that there was any inhibition, I did not count it.